Welcome back to QHSE Talks. This is your go-to guide for absolutely mastering the Certified Safety Professional exam. Today, we are tackling a really interesting and super important topic from Domain 6, Biological Hazards. Now, you might think you know all about workplace stressors, but trust me, when we're talking about living threats, the rulebook gets thrown right out the window.
So, what's the big deal? What really makes these hazards so unique? Well, it's pretty simple actually. Unlike a chemical, a biological agent is alive. It's a completely different kind of threat. And for the CSP exam, getting your head around this one key difference, that's the foundation for everything else we're going to talk about.
Let's really break this down. The two game-changing words here are living organism and reproduction. Think about it. A single bacterium can multiply into millions literally overnight. That's exactly why your standard occupational exposure limits, the ones we use for chemicals, are basically useless here. You can't just measure parts per million. We need a totally different risk-based approach, and that is precisely what the exam is going to test you on.
To control these threats, you first have to know what you're up against. You got to know your enemy. And for this exam, just knowing the name of the disease isn't enough, not even close. You'll need to know the specific agent that causes it, and sometimes even its common nicknames. So let's take a look at the major players on the field.
First up, we've got bacteria. These are some of the heavy hitters you are for sure going to see. We're talking about things like anthrax, which is caused by Bacillus anthracus, or tuberculosis, which comes from Mycobacterium tuberculosis. Being able to connect these pairs, the disease and the agent, is a classic way the exam will check your fundamental knowledge.
Next up, viruses. You'll definitely see these pop up, especially in healthcare settings. Agents like hepatitis B and HIV are pretty common knowledge, but don't sleep on the zoonotic diseases. The ones that jump from animals to people. A perfect example is ORF, also called sore mouth disease. It's a real-world risk for anyone working with livestock like sheep or goats.
Now for a category that can be a little tricky: rickettsilia and chlamydia diseases. These guys are unique bacteria that can only survive inside other living cells. The one to really remember here is psittacosis, which also goes by ornithosis. The name itself is a dead giveaway. It's transmitted from birds, especially parrots. That's the kind of specific detail that'll score you easy points.
And finally, rounding out our list, we have fungal hazards. These are often tied to specific environmental or job-related exposures. Think histoplasmosis from disturbing bird or bat droppings, or aspergillosis from working in moldy buildings. With these, knowing the source is just as important as knowing the agent itself.
So, we know the enemy. How do we fight back? Well, biological safety really comes down to a simple but incredibly powerful two-pillar containment framework. And let me tell you, mastering the difference between these two pillars is an absolute must for the CSP exam.
This table here really lays it all out perfectly. On the one hand, you have primary barriers. These are all about the human element. It's your lab practices, your sterile techniques, and the safety equipment you use directly, like a biosafety cabinet or your PPE. It is your absolute first line of defense. Then, on the other hand, you have secondary barriers. This is about the building itself, the facility's design, specialized ventilation, like negative pressure rooms, and high-efficiency HEPA filters. So here is the crucial point. If you remember only one thing, make it this. Primary barriers protect the individual worker inside the lab. Secondary barriers protect the community and the environment outside the lab, worker versus community. It's a simple, powerful distinction, and it will help you nail so many questions on the exam.
Okay, let's move on to the biosafety levels, or BSLs, and see how this all builds into a real-world hierarchy of controls. It's a pretty elegant system that scales the level of containment directly to the level of risk that the agent poses.
And this slide just illustrates that progression perfectly. BSL-1 is your basic teaching lab. BSL-2, we step it up from moderate risk agents that we see all the time, like HIV. BSL-3 is where things get really serious. We're talking about agents that are dangerous if you inhale them, like tuberculosis. And then at the very top, BSL-4 is maximum containment for the world's most dangerous pathogens, like Ebola, where we literally have no cure.
Now pay close attention here because this is a huge exam point. The single defining feature of a BSL-3 lab is its advanced engineering controls. You absolutely must know that it requires inward directional airflow, also known as negative pressure. This is a secondary barrier, and its whole job is to contain dangerous aerosols and stop them from ever escaping the lab. It's all about controlling that inhalation risk.
And at the very peak of the pyramid, we have BSL-4. This is the realm of total isolation. Think separate, dedicated buildings and full-body, positive-pressure spacesuits. But what really makes a BSL-4 agent so terrifying isn't just that it's deadly, it's the fact that we have no vaccine or therapy for it. When there is no safety net, containment has to be absolutely perfect. All right, so let's bring this all together. How do you apply this in the real world? Well, you use the classic industrial hygiene framework: anticipate, recognize, evaluate, and control, or AREC. This is exactly how you'll be expected to think on the exam.
See, this is how it all connects. You anticipate and recognize that a veterinarian might be at risk for brucellosis from an infected animal. You evaluate that risk using tools like bioaerosol samplers. And then you control the hazard by putting the right BSL in place, which means using the right combination of primary and secondary barriers. See? AREC gives you the roadmap.
Okay, now for what might be the most important part of our chat today. Let's do a rapid review of the high-yield facts you absolutely, positively must have memorized for exam day. This is the critical stuff that shows up again and again on the CSP.
I want you to lock these 3 distinctions in your memory. Number 1, biological agents are alive and they reproduce. Number 2, primary barriers protect the worker, secondary barriers protect the community. And number 3, the ultimate dividing line between BSL-3 and BSL-4 is the lack of a vaccine or therapy for BSL-4 agents. Got it?
And here are 3 more specific connections you should know cold. When you see tetanus, think soil. When you see psittacosis, think birds. And when you see BSL-3, think aerosol or inhalation risk. Nailing these little details shows a much deeper level of mastery.
So, we'll leave you with this final thought. Passing the exam is about mastering the hazards we know about today. But becoming a true safety leader, that's about learning how to anticipate and prepare for the biological risks that are going to emerge tomorrow.
Thanks so much for tuning in to QHSE Talks. Remember, every minute you invest in learning this stuff is a step towards a safer future for everybody. If you found this helpful, be sure to like, subscribe, and share it with your network. Keep up the amazing work, and we'll see you in the next one.